Okay, welcome back to another workstation of Hort Science Live. This workstation is titled Principles of Seaweed Biostimulants. So obviously we're going to explore some of the world of seaweeds, which can be used as biostimulants. Before I talk about that, my name is Richard Collins. I'm a technical manager with ICL. Work for a particular bit of ICL called AmegA Sciences, which is a research and development and manufacturing site based in Daventry in the Midlands of England. Manufacture a wide range of liquid products for agriculture, horticulture, for sports turf sectors as well, including wetting agents such as H2 Grow, which you may or may not be aware of, liquid fertilizers, biostimulants, obviously linked closely with this particular workstation, and adjuvants for plant protection products and so on. So my role as a technical manager, quite a significant bit of it is being involved in trial work, whether that's kind of initial research and development trial work, so screening new particular products which may or may not have roles as biostimulants. So I may be carrying out that trial work myself, maybe carrying out that trial work in conjunction with external parties as well, and also trial work as well to back up existing products. Enough about myself. Just go through some of the aims of this particular workstation. So I'm going to give you an overview of seaweeds and particularly their use as biostimulants. So hopefully by the end of this presentation, you'll be able to make your own mind up whether seaweeds have a role as biostimulants in horticulture. I'll talk about some of the background into seaweeds because seaweeds are quite diverse, and the way that we can extract some of the potential goodness out of the seaweeds and use them in useful products in horticulture. I guess one of the themes throughout will be the importance of research and development, obviously crucial to support any reputable product out there on the market. And we'll talk about some of the potential uses of seaweed extracts, some of their potential benefits in the production of our plants. So if we start with talking about some of the key differences in seaweed products. So there's lots of different seaweed products out there already on the market. This presentation is not designed to kind of knock any particular product, but to highlight that there are some very, very key differences between seaweed products. So I've got two bottles here. One I've called Seaweed A and one I've called Seaweed B. And it's very, very likely that there's going to be some fundamental differences between this product, Seaweed A, and this product, Seaweed B. If you were to use this on a particular plant, it may have a particular benefit, enhanced rooting, for example. But just because Seaweed B also contains seaweed doesn't necessarily mean you'll see the same benefits or any benefits that you saw with Seaweed A. And there's some key reasons for that, which I'll go through on this particular slide. Seaweed species. So there's actually many different species of seaweed out there. There's some key ones used in our biostimulants. And on the next slide, I'll talk a bit more about that. Many different extraction processes out there. So with extraction processes, what I mean by here, we're taking some of the chemistry out of a seaweed and putting them into a bottle. Hopefully, some of that chemistry will have biostimulant benefits on our plants. So different extraction processes. So we may be getting different things out of the seaweed into our bottle. Of course, the concentration of seaweed in each extract. So Seaweed A, for example, may have 10% seaweed. Seaweed B may have 5% seaweed. So you would expect this one to have a bigger effect than this one here, or a better chance of having an effect than this one here, if other things are like extraction process are the same. Of course, application rates, seaweed products out there by seaweed biostimulants should have clear application rates out there, and you would hope that the application rates on the bottle are enough to cause a beneficial effect when we apply to our crops. But there may be products out there which have low application rates and which are not going to have the beneficial effect that we want on our plants. Of course, there's many different application methods. We explore a few of those as we go along. Foliar application, for example, root drench, and so on. Some of them may have different effects to others. And what I call a co-formulant. So within our bottle, for example, we may have nutrition. More than likely, we will have some nutrition, as well as the seaweed extract. So it's important to understand that when we apply seaweed A, it's the seaweed having an effect, not just the nutrition. And good trial work should be able to iron out any differences. So all these taken into consideration, these are the key differences, or some of the key differences, which may make seaweed A stand out compared to seaweed B, and therefore have a different effect, or a effect, on the production of a particular plant. Okay, so in terms of species of seaweed used in our biostimulants, so what are seaweeds? There's not kind of one group of seaweeds. There's actually, many different species, which about 10,000 or so, which could be classified as seaweeds. In terms of our seaweed biostimulants, they tend to come from 3 different groups. One of those groups, red algae. One of those groups, green algae, and one of those groups, brown algae. By far the most common group of seaweed which our biostimulants come from are the brown algae. And this particular species of brown algae, Ascophyllum nodosum, the vast majority of our seaweed extracts, seaweed biostimulants, are based on. A little bit about Ascophyllum nodosum, found in the North Atlantic. Its habitat is intertidal, so it's going to be exposed to high tide, it's going to be exposed to low tide, and really exposed to different extremes. And therefore, this species has had to adapt to these extremes. So low tide, for example, when the sea goes out, obviously it's exposed to the air. If you think of the different seasons, it can be exposed to wide variety of temperatures. It could be out there exposed in very, very hot sunny conditions, which will be very, very stressful for this particular species. It could be exposed to very, very cold conditions as well in winter time. And obviously, not being covered by the sea, you're prone to drying out or desiccation. High tide, obviously the plant's going to be fully submerged, very, very different conditions to low tide. And obviously found in the sea is very salty, so it's got to be adapted to saline conditions. So because of this intertidal habitat, these extremes, this species and other species of seaweed has definitely had to adapt to these extremes. And these are adaptations in terms of chemistry. Can we take them out of the seaweed and put them into our biostimulant products to hopefully have an effect on our plants? When we're taking some of this chemistry out of the seaweeds and hopefully putting them into our products, we carry out extraction, and there's different extraction processes which I'll touch upon. These are very, very complex mixtures. You're not taking just one compound out of the seaweed, you're taking a wide variety of compounds out there. So polysaccharides, which are carbohydrates, minerals, vitamins, oils, fats, acids, antioxidants. Antioxidants is something that we'll touch upon later. You'll probably come across it from a human health situation, eating foods high in antioxidants, and hopefully you'll be aware that they're useful in a plant production situation as well. Different pigments, different hormones as well. So these are hormones which can have growth regulatory effects on our plants, hopefully beneficial ones. So these are very, very complex mixtures. So yeah, as I said, with our extracts, what we're trying to take out, some of this chemistry, hopefully beneficial chemistry, out of the seaweed and put it into our bottle, which can then hopefully have an effect, beneficial effect of some kind on our plants, whatever those plants are. Different types of extraction processes. I'm just going to highlight some of the more common ones out there. You may or may not have come across extraction terminology when you're looking at some product literature. you may or may not have come across water-based extraction types, acid-based extraction types, alkaline-based extraction types. Each of these are used commonly to produce products. Some may or may not be better than others, but they'll have different end results in terms of the chemistry taking out the seaweeds. Again, looking into the literature, I'm not saying every product based on utilising water-based extraction, but basically what we're doing here is that we're macerating or blending up our seaweed, and then we are kind of hydrating, mixing it with water. Some of that chemistry, some of those compounds are coming out the seaweed, going into the water-based liquid, which we can then bottle off. Obviously, we need to remove the solid residues. We don't want those to go into our bottle because that can obviously block sprays and so on. Another extract, acid extract. So basically here, again, we're macerating or chopping up our seaweed, and we're, rather than extracting it in water, we're extracting it in a solution with a very, very low pH. So we're using things like sulfuric acid, nitric acid, hydrochloric acids. Alkaline extracts, essentially the opposite of acid extracts, that we're taking our chopped macerated seaweed and incubating it in solutions containing a high pH. Potassium hydroxide, commonly used, sodium hydroxide, also potentially used. Different potential benefits of each. The literature suggests that water-based extractions, you get higher amounts of hormones, so potentially plant growth regulatory effects in water-based extracts. Acid extracts, again, another study in the literature suggesting we're getting certain carbohydrates which have plant growth regulatory effects. Alkaline extracts, what we do definitely get here is breaking down of some of these carbohydrates into novel compounds which aren't present in the seaweed but may or may not have a beneficial effect on plant growth. So we're getting production of new compounds which aren't in the seaweed but are in the bottle. On the turf and landscape side, ICL have carried out a fair bit of research on different extracts of seaweed and their effects on plants, led by Andy Owen. And we've got 3 different extracts here. I'm not going to name the different extracts, but you can clearly see here different visual characteristics of the extracts. So certainly we're getting different chemistry in each of these bottles because of that extract. Obviously, what we don't know is if that's, at this stage anyway, if that's translated into improved plant performance. We may touch upon that later. And you can analyse the extracts as well. Here we've got an NMR analysis of the extracts, two different extracts. Extract 1 here, extract 2 here. Each of these peaks represent, different chemistry within each extract, and you can clearly see extract 1 here, we've got these different peaks, which mean we've got different compounds in this extract compared to this one here, and these compounds may have beneficial effects on a plant which may not be present in this extract here. One study from the literature published in 2013 did look at different extracts, and again, I'm not going to name the extracts, 3 different extracts here. Carried out different studies in lettuce and oilseed rape, applied the extracts to these plants in non-stress conditions, so no drought, no salinity. None of these extracts had a beneficial effect in these unstressed conditions at the rates that they were tested. When they grew these plants in high salinity conditions, what they did see was that all 3 extracts did have a beneficial effect on plant growth compared to the untreated plants. But this particular extract here, extract 1, had a very, very beneficial effect on plant growth in high salinity conditions. At one of the rates, plant growth was the same as in unstressed conditions. So getting some sort of protection in saline conditions with seaweeds, with particularly this extract here. And this terminology here, antioxidants, we talked about it before. In stress conditions, whether it's drought or saline conditions, plants can produce certain substances called reactive oxygen species, which are very, very damaging to the plants. and antioxidants generally mop up these reactive oxygen species and protect the plant. They looked at the antioxidant levels in plants treated with extract 1 and extract 2 and found that there were high, very, very high levels of antioxidants with this extract, which, if you remember, performed very well in saline conditions. So, this extract is producing antioxidants in the plants, which are able to protect it very much against saline conditions. It's all looking good for this particular extract in this study, but when they exposed plants to different fungal pathogens, it was actually this extract here which performed better. We got better defense against the fungal pathogens than this extract here. We still did see a benefit, but not to the same extent here. So really is showing that the extraction process is very, very important in terms of these seaweed biostimulants. If you look in the literature and product claims as well, you can look at different benefits of seaweeds, what they can do to plants. I've kind of grouped them here into different headings, rightly or wrongly. Certain benefits in terms of plant growth and quality, which is what you would hope from a biostimulant. Some benefits are to do with nutrient uptake, so the ability of treated plants to take up nutrition. A lot of work on environmental stress tolerance, and there's work as well on defense against things like fungal pathogens, things like nematode pests. So I'll explore some of these in a bit more detail, starting with plant growth and quality. So for each of these, I'm going to talk about there are studies which back up each of these claims. Some seaweed studies have shown that you can get better chlorophyll or higher amounts of chlorophyll in the treated plants. Obviously, chlorophyll, very, very important for photosynthesis, plant health, and so on. Lots of studies out there showing you can get higher yields and quality of yield and plant with treatment with seaweed biostimulants. Shelf life as well, obviously for horticultural crops, such as fruit, such as salads, and so on. Shelf life is very, very important. So if we can improve that by the application of biostimulants, that's a positive. Nutritional quality, leafy salads, for example. It's been shown in studies that application of seaweed biostimulants can improve nutritional quality. Production of leaves, stems, and so on. Again, the studies out there which show we can get better vegetative growth with seaweed biostimulant application. Root architecture as well, so how the roots grow, but not just how the roots grow, the amount of roots that a plant is produced. And ICL have carried out quite a bit of work which show that particular extracts we can get higher amounts of root growth. One particular study which I carried out at the University Centre Myerscough looked at incorporation of seaweed biostimulants at different rates into growing media. Grew plants from seed over a 28-day period, and at the end of that 28-day period looked at the amount of shoot root growth. So just to quickly go through this, we've got 5 different treatments. We've got some plants grown in untreated growing media, so no seaweed application. And then we've got 4 different treatments where we've got seaweed application, but at different rates. Rate 1 is the lowest application rate. Rate 4 is the highest application rate. And these correspond to these graphs here. So we've got shoot biomass, so the amount of shoots at the end of the 28-day period. This dark blue line here represents the untreated plants, and every one of these bars represent the seaweed treatments. This at the higher rate, this at the lower rate. And what you can see at the 28-day period, we've got these 2 rates here, rate 3 and rate 4, where we've got a significant benefit in terms of shoots at the end of the 28-day period. So 15% extra shoots and 18% extra shoots. So, a clear benefit in terms of application of the seaweed and incorporating it into the growing media. In terms of the roots, we saw similar findings, untreated bar here, and at rate 1 and at rate 3, we got significant benefits in terms of rooting. 29% extra rooting and 28% extra rooting. So as you can see here, we're actually getting more roots being produced compared to the shoots. And this is what we consistently see with this particular extract, and that rooting is very, very much benefited Obviously, total is the roots and shoots added together. And you can see here at rate 1, rate 3, and rate 4, we've got significant benefits over the untreated control. So, clear benefits in terms of shoot growth, root growth, but also interestingly here, kind of the rate effects. I would say here the rate 3 we've got a better effect than rate 4. So, rate, as I mentioned earlier, is an important factor. There's no point going for this high rate. We're not getting any additional benefit from a plant production point of view. You're actually spending more money and getting no extra benefit from it. For the rooting trials, we can look at growing plants in particular chambers. And before, this is pansy growing in a chamber. Got growing media here with different incorporants in. Obviously, we can look at the patterns of root growth over a period of time and look at root biomass at the end of that period. And we can use pot-based trials as well. So this is a trial we've got up and running at the moment here. Taken one treatment out here of a randomized replicated trial just for visual purposes here. Still early days in this particular trial, potentially slightly more shoot biomass in this trial here. Still early days, hard to interpret that. But of course, what we don't know at this stage is what's happening under the ground in the growing media, whether the rooting's improved. At the end of this particular trial, we will be able to see that. So yeah, I carried out a lot of pot-based work in things like seaweed, biostimulants. It's a good screening method. We can look at the effects of biostimulants on roots and shoots, as I saw in that previous trial. We can look at rate effects. We can look at different application methods, whether that's incorporation, whether that's foliar application, and so on. We can look to see if we get benefits in a environmental stress. So we can dry out these plants. We can subject them to reduced watering. Does the seaweed have a better effect in drought conditions, for so on? So there's all sorts of things that we can do. Going back to these potential benefits of seaweed biostimulants, I'm not going to focus on nutrient uptake, but just to highlight, if we're getting better root growth, in theory, we should be able to get better nutrient uptake because those roots are going to be able to harvest more nutrition from the growing media from the soil. What we'll focus on a little bit is resistance or tolerance to environmental stresses. And again, there's the studies which back up each and every one of these claims. A lot of work which has shown benefits of seaweed in drought stress. And again, ROS, this is what I talked about earlier, reactive oxygen species produced by plants under stressful conditions can be very, very harmful for a plant. So if seaweed can reduce the levels of these reactive oxygen species, then that is very, very beneficial. High salinity conditions, studies out there show that seaweeds can help in saline conditions. And again, we can get these production of reactive oxygen species in saline conditions. Drought resistance, again, appearing here, and one study suggesting that we get more of a certain amino acid being produced. This particular amino acid is very much associated with resistance or tolerance to drought stress. And the studies out there, again, which have shown effects of seaweed on drought, beneficial effects of seaweed on drought, and how it can affect gene expression. So I'm not going to go into any details, but there's some really good studies out there which show the potential benefits of seaweed. This particular study here, just a nice visual demonstration of how seaweeds can help in a drought situation. So we've got two different treatments here. We've got untreated plants here, and we've got plants treated with a Ascophyllum nodosum extract. These plants were then subjected to drying out, and you can see here the untreated plants, very, very severe stress appearing by day 4, a lot of death by day 4. And the seaweed extract treated plants, although we are getting the onset of stress, you would say that is a lot more tolerant to this drying out than these plants here. So a nice visual benefit in this particular study here. And of course, they produce nice graphs. Survival rates of this particular line here of the seaweed extract treated plants being a lot higher, and the water content being a lot higher throughout that process with the seaweed extract treated plants as well. They also show that the stomata, which are obviously key areas where a plant can lose moisture, the seaweed can partially close the stomata, which can help retain that water. They showed that photosynthesis was maintained for longer during that drought stress, and that, again, these antioxidants were produced at higher levels in the seaweed extract treated plants. So quite a nice study looking at effects of seaweeds on drought stress. And one extra benefit which I haven't touched upon is potential effects of seaweeds on seed germination. And this is a trial that, again, I carried out at the University Centre Myerscough at the beginning of the year. Just to try and explain a little bit, basically we've got seeds germinated in pots, and we've got different treatments within those pots. So we've got treatment here, which are untreated, no biostimulant application. And we've got 1, 2, 3, 4, 5 different biostimulants. Some of those are seaweed biostimulants, some of those are different biostimulants. These two bars here are based on seaweed extracts, so the treatments are seaweed extracts. And we look at mean germinated seeds per pot over 9 days after sowing, 11 days after sowing, and 23 days after sowing. 9 days after sowing, you can see we've got a benefit, significant benefit in terms of germination with both of the seaweed extracts. Things are caught up a little bit by day 11, but by the end of this particular trial, by day 23, again, the seaweed extracts, we've got better germination over this time point. 23 days is quite a long time for these particular plants. I'm not going to say the plants, but these particular plants to germinate. So good overall benefit in terms of speed of germination and total germination during this trial. And we've got another treatment here, which isn't a seaweed extract, but definitely the seaweed extracts are performing well here. Similar here, same treatments, slightly different time points, but here we've got plant height per pot. And again, seaweed extracts we're getting by day 9, significantly higher plant height compared to the untreated. Again, day 11, day 14, and by the end of the trial, that plant height is consistently higher throughout. And obviously we can harvest the plants at the end of the trial, just like for that previous trial that I showed you. Look at dry weights of the shoots, of the roots, and the shoots and the roots. And both of these seaweed extracts here, we got significantly higher shoot biomass by the end of the trial. But look at here, in terms of the rooting, both of these seaweed extracts here, far greater root biomass by the end of the trial. These seaweed extracts, these two seaweed extracts are really having a big difference in terms of rooting in this particular trial. Just to touch upon methods of application, obviously different methods out there, and just be aware that on a bottle in the product literature, it should say whether it should be applied through foliar, through root drench, growing media application, through seed treatments, all different options out there. And foliar might have a different effect to rooting, for example. So just something to bear in mind there. Obviously follow what the product literature is out there, and ask questions in terms of a particular product. Is there any trial evidence to back up that particular method of application? Obviously, just to leave on some kind of take-home messages, obviously covered a lot of the fundamentals of seaweeds, and appreciate it is quite complex, but these are very, very complex extracts, and it's hard to kind of simplify the story. But from my point of view, just be open-minded. If you're not a user of seaweed biostimulants, or any biostimulants for that matter, just be open-minded about them. Before I joined ICL, I got experience of seaweed biostimulants. I perhaps wasn't open-minded about them. I was carrying out some trial work, didn't really expect seaweed biostimulants to have any kind of effect on plants, and I was very, very surprised. We did see some really beneficial effects of seaweed biostimulants. So be open-minded. Be open-minded to trialing them as well. All seaweed products are different. Clearly, different seaweed species out there, different extracts, and so on. So one product is not going to be the same as another product. So be aware of what claims back up each individual products. There's all sorts of differences out there. yeah, same thing really. Be aware of the product claims, because all seaweed products are different. So does the company selling that particular product have trial evidence to back up the claims? Check claims are supported, not just with seaweed biostimulants, with any products out there really. It's very, very important. I know ICL take a lot of pride in terms of supporting any product out there having good evidence to back up the products, so responsible advice can be given. You do try to use biostimulants for specific purposes out there. There's lots of claims for biostimulants. Be aware of ones which claim that they do absolutely everything, because that is perhaps not practical. So maybe you want your plants to get established earlier than if you weren't applying biostimulants. Maybe you're after improved rooting in the early days of production. Use biostimulants for specific purposes. And of course, as with any biostimulants, seaweed extracts, they're not a substitute for good agronomy. You still need optimal nutrition, optimal irrigation, lighting, plant protection, and so on. They're not going to make up for poor growing conditions, poor agronomy, and so on. So yeah, finally, just like to thank you for spending the time to listen to this presentation, this particular workstation. Obviously, keep an eye out for future ICL product developments, whether that is in the field of seaweed biostimulants or general biostimulants. It's important that the products out there, as I said, are backed up with particular claims, and I know ICL are very, very responsible and will back up any future developments with good trial work. So thank you for listening.